Now it's time to look at another example of gene regulation, also in E. coli, the trip operon. The trip operon encodes enzymes that synthesize the amino acid tryptophan. So there are actually 5 polypeptides encoded by 5 coding regions here. It's an operator and a promoter. So you get a polycistronic messenger that's translated. You get 5 polypeptides, and they actually form 3 enzymes. Some of these polypeptides have quaternary structure. But they catalyze the enzymatic steps that I've shown here, the reactions, that lead you ultimately to tryptophan. When is it going to be advantageous to the E. coli to transcribe this operon? Well, when there's not much tryptophan in the cell. Then it would help the organism to transcribe this, make the enzymes, make some tryptophan. When there's plenty of tryptophan in the cell, then it would be advantageous to quit transcribing this so that you don't waste energy making extra tryptophan when you've already got some. So, how does it manage this? Well, first of all, the organism regulates the transcription of the trip operon by a negative control mechanism, similar to what happens in the lac operon. Katie, that's got to be a coincidence. So, there's the trip operon. There is elsewhere in the cell a gene that encodes the trip repressor. This is a repressor protein. In its initial state, just the protein alone, it's inactive. Tryptophan binds to an allosteric site in that trip repressor. Tryptophan, by the way, is called the co-repressor. And when it binds in that allosteric site, it changes the conformation of the trip repressor, making it active. The active form then binds to the operator. When it binds to the operator, that prevents RNA polymerase from transcribing because the repressor's in the way. Typical negative control. Now you might think this is all you need. When tryptophan is high, when there's lots of tryptophan around, then you'll have active repressors. They bind to the operator, stop transcription. Great. When tryptophan is low, then you'll have inactive repressors, and therefore they won't be bound to the operator, and RNA polymerase will be able to bind the promoter and transcribe the operon, make a polycistronic messenger, make the enzymes, make more tryptophan, everybody's happy. Well, apparently, it's really important not to make too much tryptophan, because E. coli has another mechanism, a sort of a fail-safe mechanism, to ensure that this operon doesn't get transcribed when there's plenty of tryptophan around. In order to understand this fail-safe mechanism, which is known as attenuation, we need to look at the sequence of what's called the trip leader. This is a little stretch. This is the RNA here shown. This is a little stretch of RNA that's right after the promoter and right before we get to trip E, the first gene. So this is transcribed, and then the trip E, the actual gene there, starts afterward. Now if we look at this little region, you see several things in it. One thing that you see, this red area here marked 1, you may be able to make this out if you look carefully, is complementary to the purple area marked 2. In fact, the DNA that was transcribed to make those was an interrupted palindrome. So 1 and 2 could pair. 2 and 3 are also complementary in reverse in sequence, and 3 and 4 in the colored regions there are complementary to each other. So actually the DNA in the leader region has 4 bits that are all palindromic to each other and could fold up in a variety of ways. Another interesting thing about this little leader segment is that before you get to the first actual gene, the TRPE gene down here at the end, there's a start codon right here. And there's actually a little coding region here. This codes for a polypeptide of 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14 amino acids. That's a very short polypeptide. Okay, most proteins are 100 amino acids or so. So this little region, you can think of it as a little mini gene that is transcribed there. Well, let's take a look. The other thing about this little short polypeptide that you should note here, yeah, it's 14 amino acids long. 2 of them are tryptophans. That's unusual. Tryptophan, you'll note there's only one codon for tryptophan in the genetic code table. Tryptophan's not a very commonly used amino acid. It's very unusual to have, this would be 1/7 of the polypeptide, tryptophans. So you might, and of course this is an operon that is synthesizing enzymes that make tryptophan. So the presence of 2 tryptophan codons probably has some significance, as you'll see later. So like I say, region 1 is complementary to region 2, so it could fold like this, where regions 1 and 2 would, at least the portions of them that are complementary, would stick together. You have a stem loop here. And region 3 and region 4 could pair up. And in fact, if you look at that 3-4 stem loop here, 3, 4, look at that 3-4 stem loop, you'll note that that is a GC-rich stem loop, interrupted palindrome, GC-rich stem there, followed by a series of U's. This should ring a bell. Right? This is an intrinsic terminator of transcription. If the RNA folds this way, this stem loop is going to bind in the allosteric site of RNA polymerase, cause it to pause. Meanwhile, the RNA will be held on to the template strand by weak AU pairs with all these U's here. This will fall off and terminate transcription. So if the RNA folds this way, transcription is going to stop before you ever get to the first gene, which was out here. So this is a way to stop transcription early. That's called attenuation when it happens. And we'll talk later about what are the circumstances under which that'll happen. Well, you'll note I've been saying if it folds this way, can it fold another way? Yeah, it can fold another way. Here's the other way, where instead of 1 and 2 and 3 and 4 pairing, 2 and 3 fold. If 2 and 3 pair up, well, now that's a stem loop, but you'll notice it's not followed by a region with a bunch of U's. In fact, there are a fair number of G's and C's following that. So in fact, this is not a terminator. If it folds in with this 2-3 stem loop, it's not an intrinsic terminator. Transcription will continue, and you would then transcribe the rest of the operon, get the polycistronic messenger, make the enzymes, make more tryptophan. So then, let's think about what happens if, by some accident, even though tryptophan level is high in the cell and the repressors should be active and should be preventing transcription, but once in a while maybe RNA polymerase gets started, gets past it, and starts synthesizing or starts transcribing the tryptophan operon. So here it is. RNA polymerase is out here transcribing away. Meanwhile, this is a prokaryote. As soon as the 5' end here is synthesized, a ribosome can assemble on it and start translating. Remember, there's this little mini gene in region 1 here. So the ribosome, that's what this red and blue thing here is, the ribosome. Ribosome starts translating it. So there it is, translating away. There are 2 tryptophan codons in there, but remember there's plenty of tryptophan around. So that means there are plenty of tryptophan transfer RNAs around. So the ribosome just cruises right on through there. It doesn't slow down at all. Goes through there, gets into region 1, cruises right on into region 2 here in the purple. Well, once it gets into region 2, meanwhile, RNA polymerase is synthesizing stuff out here, where RNA polymerase makes region 3 But region 3 is unable to pair with region 2 because the ribosome is in the way. So region 3 is sitting around. Meanwhile, RNA polymerase makes region 4. Well, region 3 is available here because it couldn't pair with region 2. 3 and 4 pair, and the 3 and 4, when they pair together, that makes an intrinsic terminator. Remember, there's all these U's out here bound to the A's in the template strand. RNA polymerase pauses, the AU pairs break, transcription's over, and RNA polymerase doesn't finish transcribing the rest of the polycistronic messenger. So this is attenuation. It's a fail-safe mechanism. There was plenty of tryptophan, and this stopped transcription early before you would make the polycistronic messenger that would then be translated to give you enzymes that would have synthesized more tryptophan. Well, what happens when there genuinely isn't much tryptophan in the cell? Okay, there's not much tryptophan in the cell, therefore the repressor is inactive, so no problem. RNA polymerase starts transcribing the trip operon, and as it transcribes along, again, as soon as it makes the 5' end, you're going to get a ribosome assembling on that region 1 and starting to translate the little mini gene. And it gets to the 2 trip codons. It gets to a tryptophan codon now. Tryptophan is low. There's not much tryptophan in the cell. That means there aren't many tryptophan transfer RNAs available. So the ribosome is sitting there. And it can't move until a tryptophan transfer RNA comes along. That may take a while. Meanwhile, RNA polymerase is out here ahead and it synthesizes region 2. Okay, there's region 2. Ribosome's not moving into region 2 because it's still sitting there and got to get a tryptophan transfer RNA, and after it gets that one, it's got to get another one because there are 2 tryptophan codons there. Meanwhile, RNA polymerase is continuing to synthesize this. It makes region 3. Well, region 2 is still available. Regions 2 and 3 pair. Okay, as you remember, that 2-3 stem loop is not an intrinsic terminator. So RNA polymerase continues transcribing, transcribes region 4. Region 4 can't pair with region 3 because 2 and 3 are already paired. So you never form the intrinsic terminator, the 3-4 stem loop, and therefore RNA polymerase just continues transcribing on out here. Eventually the ribosome gets through here and cruises all along, and eventually it will translate the trip E, D, C, B, and A, the polycistronic messenger that RNA polymerase is going to synthesize. You'll get the enzymes, make more tryptophan, everything is good. Again, when the tryptophan concentration is high, and you shouldn't really have been transcribing this polycistronic messenger at all, the ribosome moves in through region 1 easily and rapidly because there's plenty of tryptophan transfer RNAs around. Nothing to slow down the ribosome. Gets into region 2, RNA polymerase synthesizes region 3. 3 can't pair with 2 because the ribosome's in the way. RNA polymerase synthesizes region 4. 3 and 4 pair because 3 was available, not being paired with 2. 3 and 4 pair, that's an intrinsic terminator. Stops transcription right here. RNA polymerase leaves. Maybe starts over, whatever. But you don't make the polycistronic messenger, so you don't get the enzymes, you don't make more tryptophan. That's attenuation.